Our second installment of Specific Immunity Lecture starts with presentation of antigens. Just to recap, we just talked about how B cells and T cells mature and what types of receptors they have on their surfaces. So now what we're going to do is start to put things together. What do B cells and T cells do? How do they get activated? So how do these receptors, how are they involved in what's going on with B cells and T cells? Alright, so one of the first steps in after having B cells and T cells mature is making them different from each other. How do they respond to specific antigens? Well, they have to have these antigens presented to them and presented to them in a way that they understand. So what happens here is the macrophages and dendritic cells that were involved in our innate immunity, the ones that took in any foreign invaders and ate them, the antigens that they collected from those foreign invaders can be presented to T cells and B cells. Just a reminder of what antigen is. Antigen is a substance that provokes an immune response. A shortened way of saying antigen is AG, so you might see that in some of these pictures. In order for an antigen to be perceived as an immunogen, so in order for an antigen to actually have an immune response, the substance must meet certain requirements of foreignness, so it has to be very different from your own cells. It has to be a certain shape, certain size, and it has to be accessible to the immune system. Okay, so antigenicity is just another way of saying an antigen behaves as an antigen. An antigen, so one of these molecules, can elicit an immune response. As I mentioned, there are certain requirements for antigen, like size. So this substance must be large enough to catch the attention of the immune cells that are going around initiating the immune response. Also, it has to be perceived as foreign. So an antigen could be an entire microbe or parts of that microbe, usually on the surfaces of that microbe. So it could be the lipopolysaccharide of a gram-negative. It could be the peptidoglycan of any bacteria with a cell wall. It can be the protein coat of a virus. So these are some examples of good immunogens. Whole microbial cells or viruses. There are foreign human cells that have markers on them that if they do not match up as your own cells can trigger an immune response. But typically small sugars or small proteins are not good immunogens, because your body has so many of these different sugars and so many of these different proteins that it wouldn't be able to recognize them as foreign. Now there are some things that are considered super antigens. And usually those are bacterial toxins. And again, these are things that are definitely foreign to the body. Now, the problem with a super antigen is that they're extremely potent in stimulating the immune response. They can activate T cells at a rate that's 100 times greater than a normal antigen. And while this might sound like a good thing to elicit an immune response to try and get rid of this toxin, it can actually cause serious side effects. An example would be toxic shock syndrome and certain autoimmune diseases. So this is where your immune system basically gets out of control and starts attacking you instead of the microbe that is invading you. So let's get back to how antigens are presented. Now that we know what antigens are, what types of molecules could be antigens, let's talk about how they're presented to our immune cells. Well, after phagocytosis, the antigen-presenting cells, like dendritic cells and macrophages, can process the different parts of the microbes and pick out parts of the microbes that would be good as antigens. Now, once they've processed the microbes, they've found these molecules that would be good as antigens. They put those antigens on the surface of the cells, bound to one of the MHC receptors. And again, MHC2 is the one that's primarily involved in antigen presentation. So, as the MHC2 markers are being put together, they bind to the antigen, and now those MH2 receptors with the antigen bound are on the surface of your antigen presenting cell. The antigen presenting cell with MH2 markers or MH1 markers that have antigen bound to them can then activate different T cells. MHC1 with antigen bound to it can activate T cells with CD8 receptors. Those are the cytotoxic T cells we'll talk about in a little bit. MHC2 with antigen bound to it activates T cells with CD4 receptors. Those are helper T cells that we'll talk about later. Right. Once the antigen is presented from antigen presenting cells, now the next step is, what do the B cells and T cells do with it? This is what we call antigenic challenge. Okay, the antigen presents a challenge to the T cells and B cells, and it's how they react to it that gives us our immune response. And we're going to start talking about T cells, and then we'll talk about B cells. T cells that are activated, so they've had antigen presenting to them, they start to differentiate. And we'll talk about what they're differentiated into. Same with B cells. Once antigen is presented to them, they differentiate as well. And once activated by an antigen, T cells differentiate into 4 different types. I've already mentioned cytotoxic and helper, so let's talk about what exactly they are. What do they do? Cytotoxic or killer T cells, those are the ones with those CD8 receptors. And again, they bind to MHC1 receptors that have antigen bound to them. These directly lead to the destruction of infected host cells and other foreign cells. So they're called cytotoxic, they're called killers because once they're activated, they kill those cells that they've been targeted to activate. Helper T cells, on the other hand, they help B cells along. So these are the ones with the CD4 receptor. CD4 receptor binds to MHC2 that has antigen bound to it. They activate other macrophages to go along and kill any microbes they're invading. They come along and they activate B cells. We'll talk about what B cells do, but one of the functions, as we mentioned before, is they produce antibodies. And they can also help to activate the cytotoxic T cells. So helpers can help along the killer T cells that we just mentioned before. There are 2 other types of T cells that are involved as well. Regulatory T cells, they control the T cell response, make sure it doesn't get out of control or start targeting your own cells. And then there's memory T cells that just hang out and are ready for future infection. This is showing you basically what cytotoxic or killer T cells do. And they have that CD8 receptor. If they bind to an MHC1 receptor on an antigen presenting cell that has antigen bound to it, they become activated. And then they can directly, through their differentiation here, they can directly go along and attack an infected host cell. So that means a cell that has foreign invaders already inside of it. So it destroys that host cell and it destroys the foreign invaders along with it. Now, helper T cells, there's quite a few of them, but we're just going to focus on the basic stuff that helper T cells do, just in general. Again, your helper T cell, those are the ones with the CD4 receptor. They bind to MHC2 with antigen bound to it from your antigen presenting cells. When they become activated, they can differentiate again But their main job is to go along activating B cells or stimulating macrophages. Basically, they help or they continue this immune response through activating other cells that do the job of killing the microbes. So they don't kill microbes directly, but they activate other cells that do. These are all the different types of T cells that there can be. The ones that I want you guys to know are the ones we talked about. Helper T cell in general, cytotoxic T cell, regulatory, and memory. Now once these helper T cells, are activated, I mentioned that their job is to activate other cells that are involved in killing the microbes. So B cells can be activated by another pre-committed B cell, so a B cell that's already been activated from before, or helper T cells that were just activated by antigen presentation. Again, these guys have those CD4 receptors, but when they present antigen to B cells, they present the antigen on their T cell receptors. So those are the other receptors that are on the surface of B cells we talked about, that have variable regions as well, that are able to change in order to bind to specific antigen. Okay, once that antigen is on the T cell receptor, the antigen is presented to B cells, and now the B cells become activated. So this is what's happening here. You have your, this is your B cell that needs to be activated. Okay, one way in order to activate it is to take a B cell that's already been activated and present antigen to this one. Another way to activate it is to take a helper T cell, so that's this interaction right here, that has an antigen bound to its T cell receptor. So if you look here in this picture, close up, this is the T cell receptor in the middle. It's got antigen, that's the blue thing, bound to it. It presents it to the B cell, and the B cell has one of those MH2 receptors on it that can bind to antigen. So this interaction right here of presenting antigen to the B cell can then activate it. Now when activated by antigen, a B cell divides, giving rise to 3 different types of B cells that we can have. So just like T cells they come in 4 different varieties, B cells come in 3 different varieties. Now the type of B cell that we think about the most are plasma cells. These cells, their whole job is to produce a whole bunch of antibodies and then release them into the tissue and blood. So their whole purpose is antibody production. But another type of B cell are memory B cells. And these guys, just like the memory T cells, they're ready for a future infection. Just like there were regulatory T cells, there are also regulatory B cells. These help to regulate that T cell response, again, so it doesn't get out of control and start to attack your own body. And these are the different types of B cells that can be produced. Once that B cell is activated up here, can become either a regulatory B cell, memory B cell, or plasma cell that's whole job is to just produce tons and tons of antibodies. Now, B cell and T cell, when they have antigen presented to them and they become activated, I mentioned that the B cells and T cells can differentiate into these different types. Well, we call this clonal expansion and clonal selection. Because B cells and T cells, they don't just kind of split up into the different types of cells. They copy themselves. It's called cloning. And then those clones differentiate into the different types that are needed. So clone is proliferation of a particular lymphocyte. when a cell copies itself, and so you have lots and lots of the same type of cell. All of these cells then are genetically identical. They're clones, they're exact copies. This shows you here that originally the lymphocytes that T cells and B cells come from are undifferentiated. So when they start to differentiate, they come up with all of these different versions of themselves. Okay, so with different receptors on their surfaces. But then once your immune system is activated by a certain antigen, there are some of these different cells that are needed more than others. So for example, this one right here, this one has a receptor that is able to bind to this specific antigen. So that means this particular one is important for the immune response. So this one is chosen for clonal expansion, meaning this one is chosen to copy itself and make lots and lots of these same types of cells. So that way these cells are available and ready for an immune response. Alright, that's it for the second installment of specific immunity. Hold on for the third installment.